everyone. Welcome to part 1C of Peptides and Proteins. This is the third sort of module called Chirality in amino acids. Okay, so as you can see, if we look at our roadmap, we are here at number 3, chirality in amino acids. So we're going to be talking about why we might be living in a chiral world. And just to recap, here's a slide showing alpha amino acids and how this is a chiral carbon. We live in a world with L amino acids predominantly. However, there are D amino acids, which would be the mirror image. And why is it that, one is preferred over the other? Because presumably if you do the chemistry from the Urey-Miller experiment, you would actually expect to get a 50-50 mixture, a racemic mixture. So before we talk about this, recall the Murchison meteorite that I talked about in the last module, which was this meteorite that fell in Australia, and on the surface they had identified about 12 of those amino acids. And there was actually a slight excess of L amino acids over D. So let's think about that a little bit. So there must have been some type of natural process that separated or concentrated one enantiomer over the other. There must have been some form of chiral selection. And how was that implemented? How did the universe do that? And what was the mechanism of enrichment? So there are a number of theories out there, and I'll just kind of highlight a few of them. One of them is that circularly polarized light from stars actually could have impacted enantioselectivity. So this is known as asymmetric photolysis. Essentially, what happens is that the two different enantiomers degrade at different rates. So it's very possible that the L enantiomer, for whatever means, is actually perhaps stronger and will get degraded at a slower rate compared to D enantiomers. So the two enantiomers degrade at different rates. So that's one. The other one is the selection by crystal faces, and this is using something like a calcium carbonate surface. And I'll come back to the slide in just a little bit, but what I want to talk to you is this experiment here called the Hazen Experiment, or the Hazen Experiment. So essentially, you can imagine in this prebiotic world, there would be a lot of crystalline calcite around. And it turns out if you actually immerse a crystal of calcite in a dilute solution of a racemic mixture 50-50, so 1:1 D and L aspartic acid, it turns out that these different enantiomers will adsorb onto different surfaces. So it's not an equal distribution. So on one side, you would get the left-handed amino acids adsorbing on one side, and on the other side, you get the right-handed amino acids. So this was sort of thought about in terms of selection by crystal faces, in that if you have something like calcium carbonate, they display surfaces that have mirror-like relationships. Perhaps it's possible that one face would be exposed to harsher elements or circularly polarized light, whereas the other one might be shielded from ultraviolet light or other sort of factors in the universe that could destroy them. So it displays surfaces that have mirror-like relationships. Okay, so here's a Hazen experiment that I just talked to you about. So enantio-enrichment. So if one phase proceeds forward while the other is chemically inert, then we can get enantio-enrichment. So you can see that perhaps these mechanisms could take this into place. Whatever the origin of homochirality, very likely the enantiomeric excess ratio was very, very low. So you're not going to get, from day one, all of a sudden a huge excess. But given that the universe has had time and, millions and even billions of years, it's possible that it could slowly build up this enantiomeric excess. However, once you start to build up one type of stereoisomer from an amino acid, more enantio-enrichment can occur. So it's sort of like this sort of self-directed, self-fulfilling prophecy in which you can get more of the enantiomer. There's two experiments I want to talk to you about. One is called, this is published by Angevant Chimie, it's called the Syrian Optima experiment, and then the other one's called enrichment by sublimation. That was published in 2007. So what are these experiments? So how are chiral amino acids selected enantiomerically? Is there affinity? Here's a question that researchers ask. Is there an affinity for L amino acids with each other? So in other words, do L amino acids coordinate better with L amino acids? Do they prefer their own self, or do they not care? And in other words, will D amino acids coordinate poorly with L amino acids? Very interesting questions. So it turns out that someone did something called the Syrian Optima experiment. It's actually quite interesting. They took a mixture of L-serine and D-serine. It turned out in this experiment there was a slight excess of this stereoisomer. And then what they did was they subjected this to some sort of symmetry breaking. So you can imagine maybe some light or something to degrade it. And what they noticed that because there was an excess of the L-serine, they started to form actually these octamers, these clusters in which you would get 8 L-serines together, these 8 amino acids, and it would form this octamer. And they were held together tightly through this hydrogen bonding. And what this means is that because this is now a tightly packed three-dimensional structure, chiral transmission actually allows for this to be much more stable. So if there is circularly polarized light around, sort of the losers from this process would include D-amino acids and even things like right-handed sugars. We'll talk more about that later. However, in this octamer, only the L-serine octamers, only the L-serine amino acids will form this. You can't add a D-serine to this, okay? So it's sort of like, these amino acids prefer themselves, and they will actually literally, hydrogen bond and physically bond with one another to make them sort of withstand harsh conditions even better. So the Syrian octamer experiment forms a non-covalent homochiral octamer in a mass spectrometer via electrospray ionization. So they actually characterize this using a tool called electrospray ionization. So at high temperatures, this thing was quite strong and stable. As I mentioned, it was chiral selective. It formed enantiomer samples, but not with racemic ones, and only the L-serine binds with 7 more L enantiomers. So therefore, they found that this could incorporate more than one type of amino acids, provided that all the amino acids have the same chirality. The L amino acids formed complexes together and excluded the D amino acids. Okay, so that takes care of the Syrian octamer experiment. The last slide in this module, the last experiment, is called enrichment by sublimation. So again, what they showed was that they could take mixtures of amino acids, so things like leucine, alanine, phenylalanine, etc., with low enantiomeric excess, maybe around 9%, and they could partially sublime these samples. However, every time they sublime these samples, that they heated them up to a very high temperature, the results show that they got a loss of the amino acids. However, the enrichment of these enantiomers they would rise, and they would get, they'd start 9%, and maybe the next time it would be 15%, but they would eventually reach somewhere between 20 to 80%. So this also indicates that a heat source may be sufficient for enantiomer enrichment. So I showed that experiment with the Syrian Optima experiment where 8 amino acids kind of form a cluster, but it's very likely that different combinations and different amino acids can form different structures that would make them more stable to sublimation or some type of heat source. So you can see here in this picture, you have a heat source, low excess of the enantiomer, and then you get evaporation and preference of the enantiomer in excess, and then you get higher excess of the element enantiomer, so higher enantiomeric excess. Here you have low enantiomeric excess. So it's possible that meteorites, which are, they certainly could be subjected to very high temperatures, that could also result in enrichment, and that could also be the one of the reasons why that meteorite that was discovered in 1969 that hit Australia had enantio-enrichment of L amino acids compared to D amino acids. Okay, so that concludes this unit here on chirality and amino acids.